All right, so we just finished talking about the phytoplankton. So you turn the page and you see section B, okay? And it says, answer all the questions from one of the options. Wish that was a little bit bigger. So we are going to skip to option D. So you just keep turning. You just keep on turning. Let's see. Just keep on going until you get to D. This is where I want you to go. Look at 32. So it's going to be, you're just going to go all the way basically to the back of this booklet. It's the biggest, thickest test of all of them, I think. All the subjects. It's paper 3. Remember, it's 20% of your grade. Paper 3. Paper 1 is 20. Paper 2 is 40. And the rest of that 20 we have left over is the IA. So keep going. Here we go. Human physiology. So we have some breastfed infants with this disease, rickets, the bone development issue. So these infants with rickets sometimes have seizures because they don't have enough blood calcium. Calcium is really important to regulate muscle contraction, as you know, that's what kicks it off, that calcium into the sarcomere. So we have a study here looking at maternal blood vitamin D levels and the incidence of these infant seizures. So here's nanograms per milliliter. You don't need to know that. Just remember it asks, you can write it down. 0 to 70. And these are broken into these quartiles. Okay. Highest quartile, it's kind of 4 different ranges. 1, 2, 3, and 4. And this is the mean, which is the average, right? So the average number. So just looking at this, infants whose mothers have higher levels of vitamin D tend not to have seizures compared to the group of the seizures, right? The seizure group, these infants who tend to have them, their range, their mothers have maternal blood on the lower range, okay? And if, let me give you, so for example, it doesn't really ask this, but if you want to know like the maternal blood, if it did ask the maternal blood nanograms range, anything along this, what you might do, so if I want to find the top of this range, I can look, I can measure it, and I can see it's about 70, about 73 millimeters. So I'll go over here, look for 73, about right there, and read that off. It looks like about 55, or I say 54. Okay, so I say the top range is 54. This bottom range, I look across, it looks like 8, but let's just check it. Looks like right in between 15 and 16. Took me a second on that, as you can tell. Right about there. So, yeah. 8 or 9? 8.5? We'll give it around 8. I don't know. Looks kind of like 9. Okay, anyway, but that's just an example of how you might use this ruler. Don't try to draw a perfectly parallel line, although that's better than not using a ruler. But that technique of actually measuring it and then going over here, that gives you a real good estimate. Okay, so as we can see, it looks like the mothers having higher levels of vitamin D does help reduce the amount of seizures. So what's the relationship? If mothers have lower levels of vitamin D, infants have a higher chance of developing seizures. And it wants you to deduce what's the reason for rickets in these infants. So lack of vitamin D in the breast milk. Deduction means you're looking, it's from the data. So you're looking at information and coming to an idea from that information. Okay, grounded in the data, from the data. Grounded in the data table, or data table / graph that they're showing you. All right, and not enough vitamin D leads to lack of bone mineralization, and that, and calcium uptake. Next question: a reason for vitamin D not be considered a typical vitamin. That's because your body can make it, right? Exposure to sun by the skin, your body can manufacture it. So that's it. Most vitamins you can't make by going out into the sun, catching some rays. Reason for some amino acids being called essential amino acids, those are the ones that cannot be made by the body, like vitamin D can. Gotta be in your diet what you eat. And in the intestine, those villi have villi on top of them, villi having villi, which makes it a large surface area. What's another structural characteristic of these special cells? One of them would be tight junctions. Now this is straight memory, hopefully you remembered some of this from the topic videos. Tight junctions, protein channels, which are part of membrane pumps, and a lot of mitochondria that is needed to run, making ATP to run those membrane pumps. All right, so a little bit morbid, but here we have cardiac muscle. During autopsies, post-mortem, people that had coronary heart disease, and then a control group. So these are people, it doesn't say, but no coronary heart disease. So normal, died of other things. And this graph is showing the relation between the quantity of cardiac muscle cells and the diameter. How big those cells are in the left ventricle. So looking at our graphs, catch an overview. The dark bars are the people that had coronary heart disease, and the peak, their average is a little bit lower in terms of the diameter. So you kind of think, oh, they're maybe smaller diameter, and the bars aren't as tall. So it looks like there's fewer muscle cells. So fewer cells, and it looks like they're smaller cells, fewer. So just the general opposite. So fewer for coronary heart disease, fewer, smaller. Okay, I guess that's not what this question. Distinguish between the distribution of cardiac muscle cell diameters in the CHD and control groups. So that's looking at these bottom numbers, these bottom bars. So if you look, the control group is higher percent of them are clustered in the middle right here, and that's kind of what I wrote here. Control has higher percent of middle values. Okay, the distribution, how or higher, there's more people at that average number. Another thing you can say, this is the last 15 micrometers is the biggest diameter. Or actually I guess it's 17. But you'll see, looks like the CHD patients, they go all the way up here to between 20 and 21. And also it looks like you have more in that range here, 4. So these have a wider range, more variation in diameter. They're spread out more. They just don't tend to cluster around that normalized value. So they're just kind of abnormal compared to people who don't have that heart disease. This is asking you, what do you know about cardiac muscle cells that help them in cardiac? This tissue is different based on the types of cells, how they're organized. So these cells are branched, they're Y-shaped, they're interconnected with each other, and that helps them transmit nerve signals between all the cells, muscle cells, in the heart. Intercalated, if you could remember something like that, that helps. So that's a structural feature to help connect the cells together that have transmission, and there's special ion channels as well that help get that transition going, or get those signals transmitted. And last question for this graph: why is there a delay in the contractions of the atria and the ventricles? Because remember, there's atria contract, then a split second later the ventricles. Everything doesn't contract at once. And the practical reason, there's this thick layer of tissue that slows the impulse between the atria and the ventricle. And they do travel using that AV node. And that delay ensures that all the blood from the atria goes completely into the ventricles before the ventricles contract. Otherwise, you're not going to have efficient blood. It's as efficient blood pumping if you don't go all from the atria into the ventricles before you pump, out of the ventricles into the lungs and the body. Okay, so that delay is very important for the optimal performance of those of the heart.